Hi, everyone. Good afternoon. I am utterly delighted to invite you all to the follow-up discussion session and conversation from yesterday's excellent presentation by Dr. Sarah Teichmann on inference, the nature of inference, and rather the inference of nature, cause and effect in molecular biology. And we are delighted to have as a conversant today Professor Neil Lawrence, who is the DeepMind Professor of Machine Learning at the University of Cambridge. Professor Lawrence is the inaugural DeepMind Professor at the University of Cambridge. He has been working on machine learning models for over 20 years. He recently returned to academia after 3 years as the Director of Machine Learning at Amazon. And his main interest has been in the interaction of machine learning with the physical world. And this interest of his was triggered by the deploying of machine learning in the African context, which I'm quite curious to hear more about from him, where end-to-end solutions are normally required. And he has inspired, his work has inspired many new directions, research directions at the interface of machine learning and systems research. And a lot of his work is funded with an AI fellowship from the Alan Turing Institute. He is also very well feted, has many awards and recognition. And I won't dwell on those because I'm dying to get into conversation and participate in the discussion that he and Professor Teichmann are going to have. So Neil, Sarah, absolutely delighted to have you both here this afternoon, and looking forward to a smooth, intellectually choppy, but hopefully a smooth presentation this evening. Thank you both.
Thanks so much, Priya. And thanks very much for the invite to be the conversant, and for Sarah also for her great presentation yesterday. So I'm going to open with a sort of short discussion. I won't use slides, but I wanted to riff off the opening to one of the questions, which was William Kwok said, before he phrased his question, he said, "I'm not a real scientist, I'm a social scientist." And I kind of think that there's a problem at the heart of what he's saying there. Because we're in a world where there's many, many complex things we'd like to understand and make decisions on. And yet some of them apparently fall beyond the remit of what science is capable of answering, because effectively it's not William that's letting us down, it's science that's letting William down if he feels that to be the case. And I think what's so exciting about Sarah's presentation yesterday, and what we're seeing in the sort of the shift in biology to a much more, I mean, it's always had quantitative elements, but the dominance of quantitative work in terms of scientific discovery, I think it has to be our hope that the same type of shift will occur with a sort of move towards computational and quantitative social science. And that's not, by the way, as I hope I'll highlight as I discuss, to sort of diminish qualitative research. I think it's absolutely vital. But I thought that was a sort of interesting starting point, because what's really going on, and what we saw a lot with what Sarah talked about, is the complexity of systems we're trying to be scientific about. And if you think Sarah sort of ended talking about the immune system, I mean, it's one of my favorite riffs to sort of say, well, you think we don't understand the brain, you should hear what we don't know about the immune system. And the immune system is sort of widespread across all animal kingdoms, and just an extraordinary type of intelligence. But not just the complexity of those systems, but also the answer of, well, if we're talking about inference about those systems, what is knowledge itself? So what does it mean to understand those things? And I think that also came through a lot in what Sarah was saying. And I think particularly you see it in biological work. So part of what that means is perhaps to be able to predict what might happen in the case of an intervention in that system. And you see that a lot in biological models. But Sarah actually used a number of different ways of trying to explain how these systems operate. And the first couple she used was what I would talk about as dimensionality reduction, giving you a map, as she would call it. And it's so interesting. And it goes all the way back to Gauss mapping technologies. And Gauss was actually doing it for mapping the Earth. But what we're often trying to do is build a map, which means a sort of functional approximation from a low dimensional space to a high-dimensional space in which we collected the data. And it's interesting the extent we use that analogy almost unthinkingly. Very, very often, I don't think Sarah was thinking about a sort of cartographic map when she said that. But of course, one of the fantastic projects that she's been leading is the Human Cell Atlas. So there's Atlas again. We see these analogies coming forward. And it's because there is a genuine relationship between that practice that we're mostly all familiar with, or maybe with Google Maps, we've all forgotten how to read a map, but the practice that we're mostly familiar with of an abstraction of the world. One of my favorite, I can't remember where this story comes from in the first place, but Bernard Schulkopf was the one that I heard it from, and it's not from him originally, was the notion of the country that was so good at maps. He said, well, you build them at 1 to 50,000. Well, we started going with 1 to 10,000. That wasn't good enough. And then we decided 1 to 5,000. That wasn't good enough. 1 to 1,000. That wasn't good enough. Eventually, we've gone for a 1-to-1 map.
I think that is Borges's story. Yeah.
That's brilliant. Thank you, Priya, for coming. I was forgetting I would end up there. So that's great that we've got the erosion. And of course, that's nonsensical. That's useless. The map is an abstraction, and an abstraction that is useful for some purpose. So this knowledge that we're interested in is therefore somehow contextual. For what we choose to abstract is dependent on who we are and what questions we're interested in asking. Sarah very brilliantly went on to sort of talk about, I thought one of the most remarkable pieces of work from my point of view was the work on the way the dimers and the trimers come together. And this sort of leading to this predictive, almost like periodic table of the protein structures. And I think it's sort of interesting. Again, we see the need to convert our knowledge into some sort of system that we can relate to, the original periodic table. And of course, and she talked about Mendel and Mendelian genetics, Mendelian inheritance, which is actually quite a nice, simple form of inheritance, but it's actually quite distant from the form of inheritance that we see in much of animal life. And I think there's an interesting aspect there that we are drawn to, obviously, explanations that we can understand. The bookshelf, the map, the Mendelian genetics, the periodic table. And I think there's a sort of follow-up question, but is that really the way the world is? Or is it just the windows that we can see through when we look at the world? The things that enable us to have intuition about what's happening in the world? And I don't know the answer to that question. And perhaps the answer doesn't really matter because it's sort of more of an anthropomorphic comment. But it perhaps does matter very much in the practice of science when we're practically, as Priya said in the introduction, I'm very keen on what happens when real world decision making comes in. And Sarah again showed wonderful examples of how you can use these technologies in perhaps diagnosing COVID and other diseases, and in future other diseases of the immune system. If we can use understanding of the Human Cell Atlas to understand how we're being infected, well, how wonderful would that be? But this leads me on to sort of what I think two really emerging challenges are in the domain here, which I think Sarah was masterfully dealing with, and she's at the head of her field. But these are just really challenging, and they keep coming up everywhere. And there's two paradoxes that I talk about as the big data paradox, and then I have a follow-on paradox called the big model paradox. So Sarah very nicely described how the data and model interact. And I tend to think of this as a spiral. And there's a wonderful quote from Karl Popper, which is the answer to the question. I can't remember whether he uses hypothesis. I think he uses hypothesis. Which comes first, the hypothesis or the experiment? To which I would say the model or the data is the same as the answer to the question, which comes first, the chicken or the egg? The answer is that they co-evolve. So what do we mean by that? Is early on, there is a very diffuse kind of experiment, which unfortunately some people call fishing expeditions. Well, it's a diffuse experiment that's trying to understand where should one focus. And I really dislike that term, fishing expedition. And then once you've done that initial experiment, that might inform you, which is informed by some understanding of the system. That helps focus, that helps refine your understanding of the system, and you focus down and down. And I see this as a sort of spiral moving forward. But what one of the challenges we're experiencing is we get more and more data in the world. It strikes me that we seem to understand less and less about the world. And there are many examples about this. So, for example, we could think about election predictions. It's, we're getting more and more information about who's clicking on what people are saying on Twitter, and yet we seem to be if anything, getting worse at predicting the outcome of elections. And this is quite a well understood statistical concept, how you predict the outcome of an election. So that's kind of odd. Or stratified medicine. So trying to sort of target drugs at the right population. As we get more and more information about patients, you'd hope that we could get better and better at that. And that doesn't seem to be happening. And I think the reason is the following. So I observed this some years ago and spoke about it first at a talk at Data Science Africa. As Priya said, a lot of my interests are inspired by applications in the African context. But I couldn't sort of give an answer to what I think is going on. And it was actually during the COVID pandemic that I've come up with this, my hesitant answer. And it comes from realization that the same thing happens with models. So the big data paradox, I think, is arising from the following. It's a sort of example of not being able to see the wood for the trees or tree. So what happens is we gain more and more data about a phenomenon. And therefore we get more and more interested in this data we have, and perhaps more and more focused. There's a sort of data inattention blindness going on. We become very focused on that data we've acquired. And we think we've got a very rich description, but the description is not representative of the wider system. And as a result, we end up making very, very biased decisions. We talked a little bit about bias in data. And one of the ways that, and Sarah does a brilliant job of bringing these data sets together, which is absolutely key, but not many people are doing that as well. You get this very sort of focused data set on one small aspect of the entity. You believe you've understood a great deal because you've got so much data, but in practice, you've actually just understood a lot about the wrong thing. So you wanted to understand the small ecosystem around a tree in a forest, but what you actually ended up understanding is sort of the details of how a caterpillar is operating on one of its leaves, which is not relevant to the question of interest. So you can see how that relates to this notion of abstraction in the map. The danger is you go diving in, and what's happening is people are losing their human intuition about what goes on in these systems. They go diving in and collect all the data in the world without that intuition, and they collect the wrong type of data because they perform the wrong type of abstraction. They get the map one-to-one scale in the wrong place. And very similar things are happening with modeling. And I believe we saw that a lot in pandemic modeling, in epidemic modeling, where you have people creating richer and richer descriptions of how diseases can move through the population. But if those descriptions themselves are all based on the idea that the disease is moving through touch rather than aerosol, most of the aspects of that description is useless to you. And actually what you want is in both cases, what you want is a model that's more abstract, that is taking account of less detail, but is more understandable by everyone that needs to operate on that model. And that's not just the designers of the model or the collectors of the data, but the domain experts. So in COVID, that would be public health officials. If you're saying to the public health people, you can't understand my model, then you've got the wrong model. You need to be able to communicate to them because they need to be able to make decisions true. And I think that that's something that goes beyond just the need for us individually to understand what's going on in science, but it must also be an ability to explain to other scientists or the general population about why these decisions are going on. So it's not just about individual human understanding, it's about societal understanding of what's going on. And I think that's going to become increasingly important. So I've just checked my notes because I'm going to sort of wrap up here, but see if there's anything important I've missed. So it's very good because I'm just checking, and I'm up to the point where it's important that people can relate to the models and inferences that we're making. And the big data and big model paradox are very much about losing the ability to do that. Now, science is super interdisciplinary, and Sarah also mentioned that, but increasingly our analogies that we're using in these domains are dividing us rather than uniting us. And I think I really welcome this series because I think it's an opportunity to discuss how that's coming about, because we should really be able to address this in the methods that we're delivering on. As a sort of final example, I think one of the real challenges we face is, as you go to sort of Priya's comment at the end of the talk was, well, what's the equivalent of the conservation of energy? What's the thing that we can do and integrate in these models that we know to be true? And I think that's one of the key sort of questions and challenges we face when we're doing this, because very often the symmetries we understand that should exist in the natural world are incredibly difficult to introduce into these data-driven models. But I think that often the answer is to do exactly what you saw Sarah doing throughout her talk, which is, yes, that symmetry is not embedded in the model, but you represent in that model, you represent its prediction in such a way that if there's an error, you see it. So if the thing is behaving in such way, so when you produce those maps, I mean, the mapping that Sarah's using, there was a sort of little Twitter thread on it the other day, but it's kind of notorious. It can potentially go wrong. So that part of the art in that is what brings Sarah bringing her knowledge, and certain people that work with Sarah bringing their knowledge to understanding when it's being done right and when the plot is valid. And some people don't like that type of subjectivity. They don't like that sort of interaction occurring. But I actually think it's vital, and I think we have to embrace that and better understand that. Otherwise, we are never going to do what I sort of said we should be addressing at the beginning, which is the challenge of bringing to that science, to that most complex of things, social science, which is where we would really like to be in the end. And with that, I'll stop. I think I overran slightly. Apologies for that.
Thank you so much, Neil, for those wonderful and rich set of comments. Sarah, did you want to take a shot?
Yeah, so I think Neil, that was absolutely fantastic, and this resonates perfectly with basically the way we operate as in the bioinformatics world in terms of, the questions that you say is the scientist has to decide which data to collect. And so from that perspective, actually big data, the data that's there is a hypothesis in and of itself, in a certain sense. It's not a fishing expedition because you have decided to collect to do a certain kind of measurement on a certain kind of sample, and that decision in and of itself, when you articulate it in that way, it becomes clear that there's an underlying hypothesis there, and I think that that I think is a fantastic way of describing, let's say, genomics, big high throughput biology, and so on, and that it's actually not that different from your conventional kind of microscopy experiment looking at sort of the cells, in one sample, in one tissue, or in one cell culture. So it's simply a larger scale version of the same thing, basically. And I think that's very illuminating to kind of frame it in that way.
Yeah, Sarah, I just wanted a point of clarification to ensure that I understood. And I mean, Neil, aren't you saying that this process also embeds subjectivity and a sort of a take? You have a take when you, did you imply that, or am I misunderstanding?
I think it does, but I think you can't separate yourself from that subjectivity in a lot of these more complex sciences. And I would agree with Sarah in terms of the data collection itself is somehow expressing a hypothesis, and one that potentially is very difficult to write down mathematically.
Mm. But the same is true of a traditional genetics. Why did Mendel decide to score the color on the peas? I mean, that's also, there's a hypothesis in a way. Yeah. Or, the Speeman's organizer experiment that I mentioned. What you choose to measure. The fly embryo, the Drosophila embryos. Those are kind of traditional, experimental, but in a way, our sort of era of functional genomics and big data, that's, it's often juxtaposed with that traditional way of proceeding, but actually, I think what Neil is saying is that it's not that different, and that completely resonates with me. I completely agree with that.
And I think one of the interesting things that happened, which I'm sort of fascinated by, is, and I don't know that I'm not a historian of science, but it's something that strikes me as possible, is that when statistical methods were introduced, they could answer a certain type of hypothesis, with statistical confidence. And I think something incorrect happened in that there was a section, and this is certainly not, statisticians don't feel this in my experience, but there's certain scientific domains where people have decided that the only form of valid science is to do something that you can convert into a sort of randomized experiment and come up with a p-value at the end of that. And that's had some awful effects. For example, p-value hacking is a consequence of people trying to conform in domains where they're unable to do that. And it's also had people dismiss things as non-scientific if that's not possible. Well, there's going to be many situations where we aren't, and Sarah gave excellent examples around climate, so let's get 1,000 randomized planets and try this sort of thing to see, it's just not going to happen. It doesn't mean it's not scientific. It just means that different approaches are required. And I think when the subjectivity is part of the scientist's expertise, and you kind of want to introduce that, I think. I mean, you have to be aware it's there, and you have to sanity check,
right?
And you have to debate.
Yeah, and I think often that gets confused with reliability, right? I mean, as in impacting reliability. I totally take your point about this idea of randomized controlled trials. I mean, in those kinds of ways of thinking, there is an implicit assumption that you are effectively marginalizing over all other variables that you don't even know exist. You don't even know what they are. And somehow you're making an implicit assumption about the variation in those variables. Like, they're all Gaussian in some way or something, right? So you are marginalizing. But I think what was very powerful about Sarah's description of the process that she's following is that she seems to be very aware and cognizant of what is being done at every stage. Sarah, that was wonderful. And that you were able to articulate it.
Yeah, and I think articulating the big data approach with the bioinformatics, as actually kind of in many ways analogous to traditional molecular biological approaches. I mean, I think that, again, that just, the way Niels framed it, that really strikes a chord with me, because it's true that, we decide what data to collect. We then decide how to model it, how to, project it from a high-dimensional space into low-dimensional space, and all those decisions, there's an interpretation element there, which is just like, conventional molecular biologists who would make a small-scale measurement, interpret the data, and then decide what's the next experiment. And so, in the big data, high-throughput genomics era, that's then, what's the next set of genomics data that you collect once you've made that model. But in a way, actually, the process itself is the same kind of iteration of, collecting data, interpreting it, even if you're using machine learning and it's more complex statistical models. And then once you've done that interpretation, you're then going to go on to the next set of data collection. And when I say you, it can be, the same research group or a different part of the community that's reading that paper. That's the computational, data analysis, and then, taking that into account for the next step. Mm-hmm. So it's, in a way, it's, we haven't fundamentally changed, the human, or the way we do science from a human point of view in the era of big data, high-throughput genomics, bioinformatics, and so on.
But I think you've changed the frame, though, because when you look sort of classically, those of us building, say, conceptual models in physics, right? There is reductionism is really built into those models. So we start out by saying, let's make a pared down model. Mm-hmm. And the subjectivity there, again, as you mentioned, comes from the expertise. When the expert scientist, she decides what is redundant, what is extraneous, what is essential, and pares down the model. And then, so you reduce actively, and then you slowly add on bells and whistles, and then go collect the data that would be appropriate, that would match the kind of more complicated mapping you have to do. But it seems to me that in bioinformatics, you are sort of consciously not doing that and being much more open in allowing many more possibilities. And does this lead, both of you, does this lead to more flexible science in a way? I mean, would you both think so?
So, I mean, well, one way I think physics is this unusual case I tend to think of a spectrum of mechanism from strongly mechanistic to weakly mechanistic. So what do I mean by that? I mean, strongly mechanistic, you're more likely to be seeing a lot of differential equations but there's still an abstraction in there. I mean, as I understand, Planck's length means the world is discrete, but a lot of this stuff is done in continuous spaces, right? So that's an abstraction. I mean, it's a really good one. Or if we look at statistical mechanics and we look at the behaviors of gas. So you get this sort of strong overlap between what your mechanistic model says and the way the world behaves, if you control the world in certain ways. Of course, if you start getting certain materials, then they break away from that. And I think that historically in physics, there tends to have been a focus in that space. If you go to a sort of very data-driven model, I mean, there's a weak mechanistic assumption in those models typically either linearity or smoothness. And very often flexibility is introduced. I think you can often unpick and say, as I think you're implying earlier, Priya, that a latent variable is being introduced to help. If there's something unknown going on that messes things up. So there's this spectrum of mechanism that I think if you go all the way back to sort of Gauss and Laplace, the people who were doing the sort of early physics, and as it turns out, early statistics, it was one world to them. And what's happened is these worlds have separated.
Yes.
Actually, a lot of the interesting stuff is in between. And I think that but it's such a difficult question, how much mechanism and where? And it comes back to that mapping thing. So in some sense, what physics is trying to do at that very microscopic level is build the one-to-one map. So say, no, we've got everything in here for this controlled circumstances. But in very complex systems, even the one-to-one map wouldn't even be useful because, the answer that the scientist wants to the sort of the questions that Sarah's asking about COVID is not because this molecule interacted with that molecule at some very low level. It's much more abstract than that. And I think what fascinates me is the ease with which human beings do that type of abstraction and the extent to which that's missing from our automated methods. So I went on too long there. Sarah, I don't know. That makes sense? Yeah,
no, that makes total sense. I mean, there are still sort of basic abstractions that underlie our interpretation of the data that things like what is a gene, what is a promoter, which is the start side of the gene. So what are enhancers? I mean, there are sort of fundamental models that we have in our mind that are implicit when we analyze the data.
They're a bit more subject to, I mean, I know it also happens in physics, but you always get the sense that, that's what we thought a promoter was, but actually this extra additional thing can happen that we didn't know about. Things seem to move and it's quite hard to describe in a very pure mathematical sense how the promoter is going to behave. I mean, if I go back 15 years to when I was modeling this stuff, we used to model, we used like catalyst-based models. I mean, when you hear what's actually happening, or you see, I think you did this wonderful thing, Sarah, you showed first this sort of simulation of what's going on in the cell, all these things moving around. And then you took it down to, okay, but let's abstract that into these block shapes. And then we can, and what's really interesting about that is you were taking the essence of what was important for the question you were answering. And you turned that into a set of pictures that everyone in the audience could immediately understand what you were doing. But of course, you've just shown the reality, or what we think might be the reality in that simulation before, where a whole load of additional stuff is going on. And I think there's this weird thing where we keep on getting drawn into the simple model that describes the abstraction, which gets us one aspect. But we find it very easy to forget, well, actually, it doesn't look anything like that in practice. Whereas, and I know Priya, so I don't want to, in physics, it feels like, well, it does look closer to the block. I mean, of course, it doesn't look like that, but you know what I mean.
Well, I mean, I think in physics, we have been very obsessed with sort of the determinism is really baked into what we project as our understanding of the world and the mathematical language that we have developed to make sense of the world. And it's surprisingly widely successful, right? So we are really wedded to it, which is why I guess quantum mechanics is so disorienting to physicists and everyone alike, that, this whole idea of deterministic relationships and, separation between subject, object, observer, observed, all of these things that come crashing down. And we have to move to a very probabilistic description. But, I think this is probably one of the sort of blinkers that physicists really have.
Would the biological context be more like the kinds of approaches that engineers use, for, I don't know, I mean, I'm thinking of, because a lot of the methods that we use for big data analysis in biology have actually come from engineering. So for instance, from image analysis or from speech recognition or from, these kinds of things. It's not actually the methods for big data analysis are less from sort of physics than
I think you're right. Yeah, I think you're right, because I think the kinds of methods you're using in bioinformatics are not searching for laws. You're not looking for laws. You're not looking for universals. And I think there's a way in which sort of physics articulated through mathematics is constantly looking for laws and, symmetries and sort of universality of descriptions. You're constantly looking for things that would, or new categories that would have some kind of universal cadence. Whereas that's why I think, it's a much more flexible approach. Yeah, possibly. I don't know, Neil, do you think that's more of an engineering take, as Sarah suggests?
I mean, a lot of it is pattern recognition,
Right.
A lot of it boils down to some form of pattern recognition.
Well, and I guess it depends which sort of engineering. I mean, in one sense, when you first said engineering, I was thinking, Sarah, like systems engineering, and there was this sort of great invasion of chemical engineers.
Yeah.
And what always strikes me is so interesting about biological systems that it's, well, because they evolved, they have all this, they don't have like, oh, we'll just connect that to that. It's like, whatever the system is like, oh, and if it could make use of, it did. And redundancy, the things that survived were the things that had a lot of redundancy. And I always find that so fascinating about when you work on biological systems, that side of it, because it really messes with your ability. You thought your intervention should show something, but actually what happens is some other subsystem compensates, because your idea of what a system is, these evolved systems didn't go, oh my goodness, well, I will have to have that as a nice separate subsystem. But I think then you came in later with, I think, the stuff, which I think is quite interesting. And I do think it's an aspect that, the more the machine learning, like convolutional neural networks.
Yeah, exactly.
And
more the more recent kind of electrical engineering, not the dynamical system sort of. Yeah.
Right. Yeah. So, and I think you're onto something there. And I think it also relates to this point around abstraction. I think the really big sort of advance that we've had in machine learning, which is perhaps, it's an odd advance because it's driven by data and compute, I feel, is just your ability to fit models to very large amounts of data and have some sort of automated abstraction about what's going on be emergent. Certainly I'm unconvinced in the biological domain, and I think that's a really interesting open question, but in the domain of vision and speech, where there are notions that are very familiar to us as humans about phones and phonemes and objects and images, which we find absolutely trivial to pick up as very small children. But we were having a lot of difficulty teaching machines what these abstractions were and how to interpret them. And the sort of way we've come towards being able to do that is just by showing these machines enormous amounts of data and having them figure it out for themselves. I mean, horrific amounts of data, way more than humans need. And I think that there's a real interesting thing that's kind of what we would like to be able to do in biology. And one of the things, like Sarah and one of the things I'm so excited about, the sort of data Sarah's producing now, is it's data at that scale where you think, oh, possibly we can do that with biological systems. But I think I partially feel it's an unanswered question about whether we'll be able to, Sarah. I don't know. What do you think?
Well, I mean, going back to, the days of genome sequencing, so the not the kind of functional genomics, more sort of downstream genomics that I work in, there's many more sequences out there than there are gene expression patterns or protein structures in the public domain, let's say, that the community has generated. And those data sets, the methods like hidden Markov models were incredibly powerful at recognizing patterns of sequence homology, methods in the kind of when I was doing my PhD in the mid to late 90s, early 2000s and so on. And that was, to me, that was, the first kind of where the light bulb kind of went on in terms of the power of machine learning, because they're so much more powerful than sort of conventional, pattern matching methods at learning patterns, sophisticated patterns, that are also hard for a human to see necessarily immediately, because you are dealing with huge data sets, and then it's really the machine that models them, so to speak. And so definitely those not astronomical, but large biological data sets exist, and they're growing, and they're growing in different domains of biology now. I think that's also the exciting thing, is that we're beyond that pure DNA, information at the level of DNA. We're now not only at RNA, but also cellular entities, which is sort of getting us closer to biology, physiology in a way, and away from the pure kind of informational level of the DNA sequence. And then it's basically the challenges and the excitement is analyzing that level of data that's closer to the biology, if you will, of a human tissue or an animal, physiological system. With machine learning. So that's
just had a thought, sorry, Priya, just sort of following up. I was wondering, because I think it kind of does escape. Do you think it's escaping the big data paradox? I don't know if you recognize, that as a thing, or if you agree with this notion that if we collect more and more, we don't necessarily understand more. And I don't personally, is it escaping the big data paradox? And if so, why? Because I definitely think there are definitely domains where we're not escaping it, I think.
I think part of this has to do with the fact that, as you mentioned, right, the E word, sort of the emergence, emergence of phenomena that we are not able to capture with any of the other, forms, language, description, models, and it's the power of emergence and what we learn from that. I found that very astounding, especially with, as Sarah mentioned, with sort of the early work on sort of the machine learning teaching neural networks to recognize a dog versus a cat, and all of those kinds of experiments that were happening, right? I think where that eventually led you to is, analyzing data sets and suddenly showing you some sort of what in my language would be called a hidden correlation, but you could not have, it's well hidden. It's one that you could have never anticipated at all. And so perhaps, a connection in a higher dimensional domain, going back to the language of maps. But I think the other sort of theme that I wanted to sort of get both of your takes on, and particularly Sarah, the notion of complexity, right? And, so what is the future? I mean, do we just have bigger and bigger and more complex data, and our tools will keep evolving? What is your sort of, what do you see as the prospects for the kind of bioinformatics work that you're doing?
Sorry, I was muted. For the bioinformatics always goes with the technologies and the data that's available, and it sort of evolves as a field in a way in lockstep. I think with that, that's a bit of a generalization, but that's certainly a tight correlation. And I would say genomics does have there's still a long journey ahead in terms of not only the size of the data sets, but also the domains and areas of biology that it can be applied to. And so, we talked about climate change and, hurricanes and events and so on, and that we can't perturb them. And one of the areas where genomics can contribute, or is contributing now, and sort of, I won't use the word encroaching, but contributing to that area of science is, environmental DNA and sort of surveillance of ecologies and environments on our planet. Sequencing basically not just animals, but also all pathogens, plants, how they depend on each other, eat each other, cope, have symbiotic relationships and so on, simply through DNA sequencing of a lot of ecological samples. And that's really, really relevant now also with the pandemic and infectious disease and
right. No, I think you make a really important point, that it has actually caused us to shift away from always thinking about competition to cooperation, right? There have been fundamental shifts in how we view, what drives sort of change, evolutionary change, and so on. You're right that.
And there's lots of areas of healthcare, that will, we can be on genomics can be used as a sort of early detection method, used by sequencing various samples from blood, saliva, urine. So there are lots of areas where the technologies can be deployed, in future. And they are evolving towards those areas and contributing to those areas. So basically diagnostics as a routine diagnostic, you just sequence a sample. And sort of in more, in very practical ways in everyday life, as well as in more arcane research contexts. And so in a way, what the impact that has is that biology is sort of becoming, as a science, I am speaking from my vantage point, but it's becoming a big data science, basically. But yet I agree with Neil, is that the measurements that you make, there's always a hypothesis. So it's not like you're blindly just sequencing every aerosol droplet in the world. There's still decisions, that are being made, and it's a scientific process with a hypothesis and a model and an iterative journey forwards in a spiral-like way, using Neil's analogy. I think that's really accurate. And I think it's sort of, I think that's where I see the future of this area of biology. Yeah.
I think what is particularly liberating about this direction is that, we can free ourselves from sort of this Popperian view of, it's all about falsifiability and so on, right? So we've broken free of that.
That's true, yeah.
And I think that's actually intellectually quite liberating, and very productive in terms of knowledge production. So the other sort of question that I would like to pose to both of you is, how do you know that you are generating new knowledge?
It's actually, establishing what's new when you're analyzing a big data set with machine learning is actually, that's actually part of the scientific challenge or task or puzzle in and of itself. And I would, sort of, I think that's a really interesting question, Priya, and very pertinent, because, we can gather the data, we can model it or analyze it using statistical, computational, machine learning methods, whatever, but then actually pinpointing what's the novel discovery, that requires going back to all the decades and decades of old literature, reading all of it, digesting all of it. And then the intellectual exercise is figuring out, well, actually that gene was known to be in that cell, but this part, actually this is a really golden nugget that's new, and then we need to articulate that clearly, because otherwise the community, can't understand the story or what's So you have to, in a way, there's also a very conventional kind of scientific exercise there, that requires the scientist to understand everything that's gone before,
right?
To present it, basically.
So, so Neil, my claim, sort of provocative claim, would be that what it means to discover has fundamentally changes in the data-rich, machine learning-driven era. Would you agree, Sarah?
Yeah. Well. Is it so different? I mean, it's different in the way the tasks that you perform. But, as Neil said, actually, there's still an implicit hypothesis in the data collection, also in the interpretation, computational interpretation. There's subjective elements and hypotheses that you have in terms of which models you use. And then, the interpretation of what's novel and what's the discovery is, again, that kind of, there is that conventional scholarship that comes into it, that you need to know everything that's come before, and you need to juxtapose the parts that, that's the novel contribution and so on. So is it actually a complete, change? Or is it just a new way of doing that exercise? And I'm curious to know what you guys think.
Yeah, Neil, what is your take? Yeah, I'm curious to hear your take.
I think sometimes we associate with Popper the sort of the falsification thing as a sort of very, but I think when you read him, I think he's not. I think it's the tool of statistical hypothesis things which turns that into a sort of a very oppressive tool. I don't think Popper ever means it like that. I think it's more for, I always read it like, my model should kind of be predictive in some way. And I think what the challenge that you're elucidating is, in what way is it predicted? And I think relates to your previous question about complexity. And I think also this historical view of everything through reductionism, that we should be able to get it down to this quite simple formula. And there's lots of reasons that if we can do it, that's quite nice. I think one answer to how do we know when we've done something new? I mean, yes, there's new to the scientific world, but one of the things that fascinates me is I read somewhere that until quite a long way through the 19th century, the whole of the natural sciences tripos in Cambridge was just teaching Newton, and now we learn that when we're 16.
Okay.
And then you've learned something new if you're compressing that knowledge. And I'm sure they must have learned other stuff. And I think part of that is because of the way in which we teach it and the understanding we have, that we developed, and it takes quite a long time. And that's the real embedding of knowledge. Now, when it comes to individual things, I think as Sarah says, it's quite hard. And I think that we can look to Popper, though, for the gold standard, that it has to be predictive in some way. But there is this close interaction between the human and the data because of the subjectivity in what data is being collected and the subjectivity in what the right aspect of modeling it is. And, that's the thing that I feel actually potentially is reduced in what we think of as the purist sciences, but we must re-embrace it if we're going to do the social sciences properly, which I think is a very, very worthy property, and they do it very well, but bring the scientific methodology to the social sciences. And it makes me wonder, is the answer a sort of meta-reductionism, that we can be reductionist but at a different level? The problem with reductionism is it doesn't allow for some of these things we've been talking about, because I think that we need to be able to say, no, we're putting that bit to one side. We're going to put Navier-Stokes to one side while we talk about this hurricane. Right. I don't quite know I don't know if that makes any sense.
I mean, this. So let me try to ask this question So Sarah, if it turns out that the notion, the concept, the conception of a cell is not the right conception, how would this big data actually show you that? Right? Let's say it's a blob, and I'm just giving some name, right? Blob doesn't necessarily mean an aggregation of cells, a completely different kind of entity, right? If it turns out that one of the ways in which we have chosen to represent is limited, because there's no question of flaw or whatever. It's just that it's too limited, right?
Yeah. It's not accurate.
Yeah. So how would it emerge from this information-rich landscape that you are exploring and mining? Is there room for something like that? And is that what you're looking for when you're looking for a discovery? Right. I'm trying to sort of see. Okay.
But there are. It is a good point, because there are cases where the concept of the cell isn't actually, applicable. Basically,
or useful, right?
Yeah, or useful, yeah. And they include sort of plant structures that are, continua of sort of tubes of nuclei in the Drosophila embryo. So those beautiful stripe patterns that I showed, it's actually kind of a bag of nuclei. There are no boundaries, no membranes or walls between the individual cells, and actually the same is true in muscle, where you've got these myotubes that are long nuclei. And but what it does come down to in those, so the word cell in or the way we analyze those kinds of tissues then is by boiling the cell down to the nucleus, and that's the kind of the entity then where the DNA, the information, is transcribed into the message. So you're then, the word, it's not a cell, it's that little kind of tiny compartment that encompasses the genetic information itself, and that's how we deal with that. And
I think there's
that wouldn't come out of the data. I think that there'd be a limitation. There are certain limits, and I think that's where Neo's thing about the subjectivity of the data that you collect is relevant, because you go in, and actually this kind of approach with this kind of sample, it's going to hit its limits, and then you decide, okay, well, in that case, we'll use this other approach. We may not disaggregate the individual cells. We may just cut through and study genomics in a tissue section, and collect a different kind of data. It may still be big data, but it's a different experiment. And that's where that's why I like that concept that actually the scientist it's not a fishing expedition. It's not just like a wild kind of blind sort of completely hypothesis-free collection here. It's not like it's a bit different from the sort of we're listening for aliens kind of thing with our radio telescopes. It's more, there's more direction.
It's a fishing expedition, but you understand where the fish should be.
Yeah.
Right.
I mean, you have an understanding of the environment
idea.
and where they hang out and where they're likely to be laying their eggs.
Right. You have a good idea. Yeah, where the ponds are. You're looking for koi. You know where the ponds are. Right.
Yeah. And I think that there's another way in which maybe another way of looking at your question, Priya, would be, going back to the map analogy, is there ever a way in which motorways don't exist? Well, yeah, if you're walking on, the Lake District, you're not so worried about where the motorways are at that moment. And I think that's where you see the subjectivity in modeling coming in. And these are sort of concepts. And we see that in physics as well, depending on what we're trying to teach, whether we're trying to teach Newton, whether we're trying to teach quantum mechanics. Our model and our understanding of what an atom is changing because you don't need to look at it with that final level of granularity. So I think there's an argument in terms of that, perhaps the cell doesn't exist when we're talking about different organs in the body. We're doing organ. Of course it exists, but it's not relevant in the way that we're thinking about the decisions that we're making, or it's only relevant at the edges. And I think there's this wonderful. Everyone loves the George Box quote, all models are wrong, but some are useful. And the way everyone uses that quote is they then describe a model that's wrong. They don't ever describe why it's useful. But there's a better bit in that, the whole paper's great, but there's another bit in that paper he says, it's important to consider in which way your model is wrong. There's no use worrying about mice when there are tigers abroad. So I think that that's the essence there, where the cell does disappear. Sometimes you don't want to take into account the cell because of you're either at such a low level that you're in the nucleus and you're worrying about something there, or you're at such a high level where it's sort of the level of fidelity isn't there. And I think that that's this acceptance of subjectivity in the modeling just comes through so strongly in biology. I think it is there in physics, but people kind of deny it. They sort of think of their model closer to truth.
Right. I mean, I guess the ambition in physics is somewhat different, right? The ambition was to always get down to the fundamental building blocks, right? So, finding out about the quarks, after having, cracked the atom and so on, going all the way down to quarks, that was the ambition, about going to what are the fundamental building blocks. Even though we know that in order to understand, the motion of this, our moon or planets in the solar system, it's utterly irrelevant how many quarks there are and what flavors they are, right? A description of that level of the system, it comes back to the mapping and your analogy with, if you're just walking along footpaths in the Lake District, you don't need to know where the motorways are, right? And I think that maybe the difference, I'm really trying to understand, as you can see, kind of trying to grapple. One of the ways, physics and biology are two different ways of knowing the world. And how are they different? Are they different? I think they are different. And I'm trying to get at how they are different, in a very basic, in the most sort of boiled down way. So what is really different? And
one of the ways I sometimes summarize it, I have this ML and the physical world thing, and I use Conway's game of life.
Yep.
And I sort of say, because I use quotes from Laplace that say that Laplace determinism, which, by the way, his whole book was about probability, and that determinism, you can't get away with it. So it's so weird that that's associated with him. But Laplace determinism, the Superman quote, if an intelligence could understand where everything is and all the laws of nature, we can predict the future. And then I show Conway's game of life, which has just 3 very, very simple rules, and point out that there's something called a loafer, which is an emergent phenomena in that game that wasn't discovered until 2013. And the effective god of the game, Conway himself, didn't know it existed. So when you go back and you read these lovely Hawking quotes, for them we would know the mind of God. Well, but you know the mind of God before all this computation has to follow from those physical laws. And there's sort of 3 ways in which uncertainty can creep in that really comes through in that Laplace quote. One is because he says would know the situation of all beings in the universe. So that's the data, the natural laws by which they're governed. So that's the model. And then, of course, the intelligence has sufficient, it's a sufficient intelligence to subject this data to analysis, he says, or something like this. And that's the compute. And one of the things with biology, it is so distant down the compute in terms of to reconstruct what's happened. And then when you consider evolutionarily, that we're actually seeing the consequences of billions of years of evolution as well, in order to sort of compute this. I mean, the whole thing with Conway, and what took us, because you have to do all that computation, all that exploration of a very relatively simple thing to find these emergent phenomena. And I just think that physics is quite close to, what are the underlying things going on with these pixels? Why are they disappearing in game of life? And biology is sort of saying, okay, that's nice that that's going on, but they've shown that the game of life is Turing complete. So this thing can compute anything. Why is it doing that, and when does it do that? I mean, that to me is a really major difference.
Sarah?
Yeah. So, yeah, I try to always see the similarities, and I'm 20% in the Department of Physics at the University of Cambridge in the Cavendish Lab in theory of condensed matter, where you have, at a sort of sociological level, you've got the theorists, which is the group that I sit in, you've got experimentalists, and in many ways there's the same kind of dynamics that you have in biology. Having said that, I mean, even a living organism like the E. coli cell that I was showing in that simulation that Neil kind of remembered with all these jiggling protein complexes, it's got 4,000 genes. That the number of interactions is kind of, combinatorially explosion of that. And the complexity of it is just, it's a different level. You've got, these circuits, but a lot of hugely feedbacks and robustness, redundancy, and all these things that we've discussed kind of built into the system. And that is different from, let's say, a crystalline material, water, ice, these materials that have been studied for decades and modeled and so on in theory of condensed matter. And so that's in fluid dynamics and so on. I mean, that's really different. I mean, it's the level of complexity, the number of components, the number of interactions, and so on. And I think ultimately that's what determines.
Well, and then going back to sort of, the protein folding problem, underneath it, you've still got all that complexity about what the interactions are in the molecular bonds that cause them to fold as well.
Well, if you take protein folding, the number of different atoms in one single 100 amino acid polypeptide chain is like it's 2,000 or so, and then they can all be interacting in different ways with each other, and that's why that problem still isn't solved.
But there's still some, to draw out the similarities, and then I'm going to sort of talk a lot. It's not work I did, but Bing-Ching Cheng, who's one of the fellows in our Accelerate program, she's looking at trying to understand the behavior of water under extremely high temperature and massively high pressure. And she ends up in this problem of, trying to do all the sort of, the Schrodinger equations. I don't even remember all the sort of details of what she's sort of doing at the low level of what's going on in those bonds. And she has to simulate it. And she's still in that position of not actually being able to do sufficient compute to simulate this very simple substance that Sarah said, very well studied. But apparently one of the things that she said that just blows my mind, this is the state that most water in the universe is in, because it's mostly in the heart of very large ice planets. Or gas giants,
okay.
And we don't even understand the phase that this water is in, and whether it's superconducting and all, which I think the implication it is. And it's for the same sort of reason that you can't do that simulation at real speed. And she ends up doing the same type of thing that I think is possible in many of these things, is replacing the actual complexity of the underlying physics, which with an abstraction that captures the salient part for the simulation you're doing. And I think that that's a commonality everywhere. That's what I think of when I think of machine learning in the physical world, the ability to find the salient part of the system that you're looking at and abstract it into something where you can compute that much quicker so that you can actually do your simulations to predict the thing you're interested in.
This has been fascinating. I've been very selfish. I think we should open up to the floor to see if our audience has any questions. So I think given that we had a very tight assembly today in order to ensure that the proceedings were smooth, people can unmute themselves and ask questions if they like.
Priya, sorry, it's Ty. The only problem is that we may have editing problems with that. I don't.
I see. Okay, we should still go with the chat. Okay,
we can. If you don't mind, I'm sorry.
Oh, not at all. That's fine.
Thank you.
Now, please go ahead and type any questions that you might have in the chat, as per usual. And I guess as we wait for people to start framing their questions, one aspect of these biological systems that I find incredibly fascinating, which again is very similar to the kinds of systems that I work on, is the temporal scales over which these systems have functioned, optimized, and operated, right? Sort of evolutionary time scales. And I wonder when, as you are uncovering these sort of complex relationships, is there interest in understanding sort of the evolutionary pattern? And I mean evolution not in Darwinian, but the changes over time, like that along the temporal axis.
Yeah, I think that's a really nice analogy between biology and astronomy, cosmology, and so on. I mean, there's a lot of. I thought we would talk more actually about temporal dynamics and correlation and causation, because that's one way that you can get a better handle on causation if there's a temporal sequence. So modeling dynamics is in biological systems, and I showed the flight development as one example. There's basically a progression happening there from the early embryo through development. That's that's a really important element of genomics and data models and so on and so forth. It's, I would say, the evolutionary sort of, evolutionary dynamics is a bit of a different problem. So if you're thinking about Darwinian evolution, because of course everything that we see nowadays is kind of a snapshot of the current time, and it's only things like digging out woolly mammoths and that kind of thing that can allow us to actually get an idea of what happened before the current time, kind of in evolution. But it's a fact that's also fascinating.
So there's a related question that Rick Crum, our evolutionary biologist, has put in the chat, and his question is: I wonder whether systems biology based on single cell transcriptomes is ready to investigate the agency of tissues, signaling centers, electrically coupled cell aggregations, and mechanical forces in organismal development.
So signaling for sure. I mean, in the sense that, I showed basically predictions of cell-cell communication through sort of receptor ligand complementarity, and you can then link that downstream to signatures of expression downstream of a receptor. You can link it to. This
is with your COVID example, right? This was
the placenta, the maternal
placenta, yes.
And how the signaling takes place between the, how signaling takes place between the fetal cells and the maternal immune cells and prevents rejection.
Right.
Basically. So there's a huge signaling circuitry basically that ensures that's very robust and redundant, actually tons of mechanisms there to ensure that tolerance and support of the placenta. I mean, I didn't go into all the molecular details. So that's certainly, single cell transcriptomes are very informative there. There are also methods that couple the single cell transcriptomes with electrical signals. Patch-seq is a technology where you couple the patch clamp of a single cell and measure the electrical signal, the conductance, and then link that to the transcriptome by then sequencing the content of what's inside the cell that you suck up in the pipette. And mechanical forces, I would say, there are various ways of that I think are very exciting going forward to link kind of mechanical properties of cells to the single cell transcriptomes. And one is to read out the molecules that determine the mechanical stiffness of a cell or the morphological properties of a cell, and link the images that you have of a tissue section in terms of the morphologies, the shapes of the individual cells, to the transcriptomes that you sequence from each voxel, each basically spatial, XY coordinate basically in the tissue section. So there are various ways of getting at these properties by coupling the single cell transcriptomes, as well as integrating other kinds of data. And I think so. It's a great question, Richard. Thank you.
Yeah, and we have a question from Jenny. who asks that yesterday you said that the predictive power is reduced with an increased amount of data. Is this really true? Isn't it rather possible that the predictive power of a model is reduced at the scale that we are currently looking at it, but may still be strong on a more detailed level?
No, so I think that was a misunderstanding. So the predictive power of the modeling is definitely increased with increasing amounts of data. There's no question about that. The plot that I was showing was data on the x-axis and knowledge and expert knowledge input on the y-axis. And basically on the left-hand side, if you have less data, you need to input more expert knowledge to interpret that data. On the right-hand side, with the high kind of data on the x-axis, tons and tons of data, the modeling, the signal is so strong
that you don't need as much
input. Yeah, exactly. Yeah, that's what it was trying to say. I hope that makes sense, Jenny. Yeah. And sorry if it was, great.
So, and I think there's a question from Ferdoz. And he says, can your work as described yesterday in in either part 1 or part 2, could that shed light on DNA double-strand breaks and repair? And, and his question is from the context of, harmful DNA damage caused by ionizing radiation. And I recently came across the following article and summarized: double-strand breaks are a type of DNA damage that can be repaired by 2 major cellular pathways: non-homologous end joining, during which the insertions or deletions may be added at the break site, or homologous recombination, in which the DNA sequence often remains unchanged. Conditions in space. I guess he's referring to a lot of the data we got out of Scott Kelly's time and the length of the telomeres and, the changes, the sort of molecular level changes from exposure to radiation he had from his long sojourn at the International Space Station, right? And so he's asking whether the CRISPR-Cas9 gene editing system offers a model for the safe and targeted generation of double-stranded breaks in eukaryotes. And would a CRISPR-based assay for DNA break induction, so he wants to know if CRISPR-Cas9 would deliver.
So I would sort of say that the transcriptomic readout of a cell can tell you about mutations only in the regions that are transcribed, obviously, and you also need to have a reference to the non-mutated reference, basically. So that can be another cell, or it can be a reference DNA sequence. And in that way, by aligning and comparing data, you can make inferences about mutations.
But can you fix them?
And well, you can, I guess the way, so cell-based therapies for instance, rare diseases, for cancers, or for rare genetic disorders, they are kind of fixing mutations by delivering cells. Well, you could either use CRISPR, yes, to fix the mutation, deliver the enzyme with a guide RNA to the cell, or you could actually deliver kind of substitute cells that are healthy, basically.
Okay.
Yeah.
I don't think I'm missing any questions yet. Please feel free to have a few more minutes before we wrap up, because we could keep going. This has been such a wonderful session, but it's getting late for Sarah and Neil. And are there any further questions? Ty, did I miss anything?
I didn't see any, nope.
Great. So I guess before we wrap up, I would like both of you to tell us briefly what are the next exciting steps in both of your research directions. Very, very curious to know what it is that you're really thinking about right now and what's very exciting?
Well, I think from me, basically the the map of all the tissues in the human body, and this, I think, really interesting from our discussion is also, what's the right level of mapping the human body? We obviously have a pretty good knowledge of our organs, but a much less accurate knowledge of the cells in our body, and actually, what are the ways in which that very highly resolved single cell knowledge of full molecular breadth, what are the ways in which that in which that is informative, and what are the ways in which that's not? So the Human Cell Atlas project, where are the areas where it can really impact on human health, on understanding, or where are the ways that actually a higher level map, a more coarse-grained map, or a more detailed map, going down to the atomic level, are actually better descriptions of our body. I think those are really interesting questions. For me, applying the power of these highly resolved and spatially resolved genomics technologies to the body, the immune system, the components of the immune system in our body is going to be, the direction of travel for the coming years, and expanding that to the human genetics scale and understanding, how we vary from each other across human beings across the globe. We're obviously different from each other, in subtle ways in this group, and there's more diversity on Earth as a whole. And can we understand that at the cellular and molecular level using these highly resolved technologies, and gathering even more big data on making a cell atlases of lots and lots of tissues of hundreds and thousands of people, and interpreting them with Neal's Gaussian process latent variable models or other machine learning methods, I think is a really exciting way forwards in this whole paradigm that we've discussed, where we use genomics technologies that generate big data sets and then use statistical computational machine learning methods to interpret them and iterate on that in this, let's call it a new paradigm of science. Whether it's really completely new or not, I think we've discussed that here, and it's been really wonderful and fun to chat to you both about that.
And Neal, what about you? What is the exciting?
One of the pleasures I get to listen to the sort of things Sarah's going to do and think, and maybe I can help. But, and that is a really great pleasure, and something I really enjoy, and I think comes in terms of immediately achievable aims. There's a lot of projects. It's one of the great things in machine learning like that. Sarah's probably the most interesting. But I think there's another thing I'm interested in and don't really have a handle on how to attack, and maybe this is like a 10 sort of 15-year thing, but I think that there's sort of a smooth path. I think it's a serious problem, is that as we've talked a lot about how these methods are used in computational biology, but of course these methods are being deployed across society. And when you think about them in the sort of social context, naive use of these methods has led to all sorts of problems, filter bubbles, manipulable elections, and so on and so forth. And this leads to a thought for me that is, we cannot be in a world where we are being operated on by the whim of these techniques. And the only way we can turn that around is by gaining more control over what these things are doing. And what does that mean for sort of normal people? What does that mean to produce techniques that normal people feel in control of, that are dictating things about how their lives are carried out? And I think that's a very difficult question. And there's sort of easy engineering answers and sort of very difficult sort of answers about what that could look like. So that's something I'm very, very interested in. But it's actually extremely hard to publish stuff in this area, and it's extremely hard to get everything in place that you need to do work in this area. So that's why it's great. In the meantime, I also get to work with Sarah's team, or my team works with Sarah's team on some of these things that we can perhaps answer more immediately.
Great. Thank you. Thank you so much, both of you, for absolutely fascinating conversation today. And Sarah, once again, thanks for the brilliant talk yesterday. And you're a very patient composer through the disruptions as well. So I just want to say thanks to the audience. Oh, I think there's a question that has just popped up. Let me, oh, this is Tyler. Tyler, who says, Jose Ortega Gasset argues fascinatingly that the phrase missing the forest for the trees is fundamentally incoherent. Forests he offers don't actually exist out there in the nominal world, but instead represent the sensation of being confronted, the inscrutable vastness of a grove of trees. To understand what a forest is, in other words, is precisely to miss the forest for the trees. Is there a sense in which Neil's big data paradox is a feature and not a fault?
I think it's a great question,
and
it goes right to the heart of what we're saying.
Yeah.
I think that the problem is we don't have control over that.
Yeah.
So I don't think, I would disagree that it's fundamentally incoherent. And I think missing its coherence is misunderstanding this point about abstraction. What is important, if I'm the Forestry Commission working out how much wood I'm going to grow next year, it's my number and scale of forests, perhaps less the individual trees. It depends what I'm doing. And this is kind of what I mean about meta-reductionism, that it's subjective, the answer to that. The problem we face with the big data paradox, and it is something that technically I am interested in how you solve it, and actually Gaussian process latent variable models do offer a solution, this sort of thing that Sarah is using in her team, is that you miss this ability to control whether you're looking at the forest or the tree. You're like, classical statistics gives us the forest. We understand very well how it gives us the forest and why. And then we think we're going to get a little section of the forest, but we go immediately all the way down to a leaf and some insect on it. We don't manage to get to the sort of the structure we're interested in because we don't have the sort of dial to move around these things. So I don't think it's incoherent in a statement, but I do think what you want is the ability to move smoothly across those spectra. And you kind of want that across all sciences, and it relates very closely to this, I think that those spectra is kind of like between physics and biology and things like that.
Yeah, and I think let's close with that very, very nice question. And I think, I'm just very tempted to make one small comment, is that, often they have been sort of, the sociologists of science have often critiqued narratives of science that scientists offer, in which they separate out the context, and the context doesn't really matter because of this sort of slim notion and understanding of objectivity. And I think that the context really matters. And I think both Neil and Sarah are telling us that the kinds of questions you ask and what question you ask really depends on the context, and what is it that informs the question is almost as important as the question itself, right? So thanks everyone for this wonderful session, and thank you both, and see you all at the next month's colloquium. Bye, take care, and good night.
Thank you, Priya. Thank you, Neil.